righty. Questions before we get started today? Oh, that was good. All righty. I have some terrible jokes to begin with. All righty. These are awful. Did you hear about the chemist who was reading a book about helium? She couldn't put it down. Two ropes go into a bar. The bartender says, "Get out of here. We don't serve ropes in here." The ropes go outside, and one says to the other, "I have an idea." He ties himself up and messes up his hair and goes back in. The bartender says, "Hey, no ropes." The rope says, "I'm not a rope." The bartender says, "You're not a rope?" "Nope, I'm afraid not." What do you get when you cross a potato with a sponge? I don't know, but it holds a lot of gravy. What's orange and sounds like a parrot? A carrot. A young woman brought her new boyfriend home to meet her parents, and they were horrified by his greasy hair, his tattoos, his dirty language, and his air of hostility. After he left, the mother said, "Dear, he doesn't seem like a very nice person." And the daughter said, "Mom, if he wasn't a nice person, why would he be doing 500 hours of community service?" Hey, how are you? Good, good, good. We're meeting in here today just for a change. All right. Now we're going to continue on and talk a little bit about proteins today. We were looking at the different structures of proteins, and we learned that proteins are divided into 4 levels of organization. And we've already looked at the simplest one, which is just what we call the primary structure. We also looked that Linus Pauling described a very, very common type of structure we call a secondary structure, and his secondary structure that he won a Nobel Prize on was the alpha helix. This is a delicate coil. It forms readily. What's happening, if you remember, is the peptide bond has one region that has a partial positive and one region has a partial negative, perfect for the formation of hydrogen bonds. So what will happen is one amino acid will go and form a weak bond with one 3 steps down. And the next one 3 steps down, next one 3 steps down. And you get this beautiful little coil. Certain proteins that are rich in alpha helices are quite stretchable. If you think about a phone cord, that you could stretch that quite easily. Very similar if you look at certain proteins, for example, wool and hair are quite flexible in and of itself because it's got a lot of these alpha helices. Now there's a second secondary structure that's fairly common. And it's the one that Thomas last time picked out. It's this one right here. These are all alpha helices, and you can see our little double arrows. What this is a structure called a pleated sheet. Sometimes you'll see it referred to as a beta sheet. It looks like an accordion, or it looks like what you might find with pleated pants or a pleated skirt, or even one of those foldable fans. The interesting thing about it is, oftentimes you know that proline, the amino acid, will go and form a little bend. Sometimes that'll form a little hairpin turn in a polypeptide. Because the two polypeptides are close to each other, they can go and instead of one amino acid and another amino acid going and forming a hydrogen bond, it'll reach across the aisle and form a hydrogen bond with something on the other side. So oftentimes when these have a little fold or we have two polypeptides that are close to each other, rather than forming an alpha helix, they will form a pleated sheet. Now, unlike an alpha helix, which is, as a coil, very stretchable, these are not. Secondary structure, in this case the pleated sheet, is very strong, but it just doesn't stretch well. In fact, if we look at things made out of silk blouses, silk ties, all right, silk is very strong, but it just doesn't stretch very well. And I love seeing it because, as you can see, it has ridges and valleys and ridges and valleys and ridges and valleys between the two. So a very, very common type of secondary structure. And when we look at proteins, you'll see that pretty much they're composed of just these two. So here's a little ridge, a valley, ridge, valley, ridge, valley. Here's an antibody. This is a defensive protein produced by white blood cells. And as you can see, in our ribbon models, it is represented by parallel lines, usually one going one way, one going the other. We'll learn that that's often called anti-parallel. But this particular antibody, as you can see, is very rich in pleated sheets. So that turns out to be our major secondary structure. When we look at a tertiary structure, this is the third level, you'll notice that it's an aggregation of secondary structures. All right. For example, here's a folded protein. And if you look at it, we have some alpha helices, we have some pleated sheets. All right. We have some regions that are neither. All right. This is what we describe as a tertiary structure. Now, tertiary is a fancy word for third. So this is our third level of organization. When we talked about all the bonding capabilities of those different amino acids, the hydrophobic, the hydrophilic, right, the acidic, the basic, and the cysteine, those are important because they dictate the tertiary structure. So if you look at secondary structure, it's really only composed of two types of bonds. You have the peptide bonds that go and hold the polymer together, and then you have the hydrogen bonds that form the alpha helix or the pleated sheet. The tertiary structure is even more folded because of the bonds between the R groups. Now, here's a great little picture kind of showing you these different types of bonds. Now, if you remember, we talked about, for example, cysteine. Here's cysteine right here. Here's another cysteine. If we have ones that are fairly close to each other, they will often go and form a strong covalent bond called a disulfide bridge. This is stuff that we found, for example, in insulin. This one is very, very common in our hair. Right? We'll talk a little bit about how proteins unravel, and sometimes we do that on purpose by breaking these bonds. If you've ever smelled a perm and that sort of stinky rotten egg smell, that's because a lot of these bonds here, these disulfide bridges, are broken, releasing that sort of sulfur. If, for example, we have ones that are hydrophilic, right, they often go and form a little hydrogen bond, and so they can go and join that. Here's a great one. In fact, you'll notice that this is acidic. It has a little negative charge. And then if you think about our rules of folding, Acidic ones bind to basic ones. And here's a basic one. This is a great example of what we call a salt bridge. It's an ionic bond joining a basic or positive amino acid with a negative amino acid or acidic amino acid. Now, believe it or not, the hydrophobic, which we think doesn't really bind with anything, it's usually found in the interior of the protein, can form a very, very weak bond with other hydrophobic amino acids. And this type of bond has a very strange name. It's called a van der Waals attraction. If you thought hydrogen bonds were weak, these are super weak. But they're enough to actually go and hold some of these little proteins together, especially if there's a bunch of them. And you can kind of see, here we are in the center of a little protein. And of course, all the hydrophobics are in there, and they can help stabilize the structure. So all these different groups, these R groups, all have their different binding capabilities, and they actually go and produce the folding, the tertiary structure. All right. So we've looked at the primary structure. We've looked at the two secondary structures. This is a tertiary structure. All right. Now, if you're a protein that's built from just a single polypeptide, all right, this is as far as you go. But we know that there are a lot of proteins out there that are built from more than one. A good example is hemoglobin. When we talk about hemoglobin, we'll see that hemoglobin is built from 4. If we look at insulin, we know that it's built from 2 polypeptides. That's another level of organization, and that's what we mean by a quaternary structure. Quaternary actually just translates as fourth, the fourth layer. So if we look at something like hemoglobin, and this is hemoglobin right here. This carries oxygen and carbon dioxide in our bloodstream. And as you can see, it's composed of 4 polypeptides. We have 2 alphas and 2 betas. Right? And they're all held together as a single molecule. So each of these polypeptides is what we call a tertiary structure, but combined into the entire protein, that's a quaternary structure. So you've got to have more than one polypeptide to have a quaternary structure. Now, what holds those polypeptides to each other? Well, it's the same as what we see with the tertiary structure. It's bonds between the R groups. So those polypeptides are joined together by hydrogen bonds. They can be joined together by ionic bonds or salt bridges. They can be held together by cysteine disulfide bridges, the covalent bonds, and they can also help stabilize their structure with some van der Waals attractions. Here's another good example. Last week I was showing you collagen, which is a very, very strong protein. And it looks like a rope. And if you look at the polypeptide chains, there's actually 3 of them. So this is an example of a spectacular quaternary structure. It has a quaternary structure. So we have 4 levels, and I wanted to show you some of these different little proteins here. This is a nice one. This is called somatotropin, or human growth hormone. This is actually secreted by a little gland at the base of your brain called the pituitary gland, but I just love it because it's kind of half and half here. You can see that we have some bunch of alpha helices on one side and then a human pleated sheets on the other. In anatomy and physiology, we just talked about this one today. This is ferritin, gorgeous, gorgeous protein. And this is a storage protein. It holds iron. Iron, which is absolutely necessary for hemoglobin to hold on to the oxygens, gets recycled. Most of our iron is in hemoglobin, but a lot of it is stored for safekeeping in places like the liver and the bone marrow and the spleen. And because iron by itself, the iron ions are toxic to cells, they have to be stored in sort of like a container. So kind of think about this as a container that holds and stores this iron so it can be used to make red blood cells. But the iron actually goes and fits right here in the very center. But to me, it looks like a fantastic, almost like a Celtic knot, or if you're a fan of Chinese checkers, it's one of the prettiest proteins I've ever seen. It's just called ferritin, the protein that holds iron. Okay. I want to talk a little bit about how important proteins are, and particularly the sequence and shape of proteins when it comes to disorders. And we're going to give you a classic example of a protein that, when it is folded wrong, can cause a serious genetic disorder. And so, and this one actually comes from looking at red blood cells. Now, if you look on the left, this is normal blood. This is normal red blood cells. Right? Now, red blood cells are incredibly common. If we look at a cubic millimeter of blood, we find that there are 4 to 6 million of them in every cubic millimeter. So there's a zillion of them. And every second, your bone marrow is making 2 million new red blood cells. So every second, 2 million new red blood cells. Now, if you look at a red blood cell, a normal red blood cell, you'll notice that when you look at the cell, you have a little clear spot in the middle. And the reason for that is, if you look at a red blood cell, you'll see it's kind of like a donut. It's been sort of squished in the center. Now, there's a reason for this. This shape, which we call biconcave, gives the red blood cell more surface area relative to volume. So it's easier for gas exchange. So oxygen will go and bind to the hemoglobin on the inside, and it will be released from that as well, and CO2 will do it too. So this provides a lot of surface area for gas exchange. The other thing about them too is that they're very flexible. If you look at films of capillaries where the blood is moving there, sometimes capillaries are so thin that the only way that a red blood cell could move through them is if they bend. Sometimes they're just in single file as they go through. Now, if you're wondering about the other little dots, that's a white blood cell here called a lymphocyte. And the little purple things, those are platelets. Those are the clotting proteins, or the clotting cells of the blood. Now, if you look over here, you'll notice that we have a lot that sort of look like this, but then we have these strange shaped ones. This, of course, is sickle cell. These are sickle cells. You'll notice that the shape is a little bit different, whereas this is very, very flexible. This is stiff, very stiff. The other thing, too, is the lifespan is different as well. The lifespan of a normal red blood cell is about 100 to 120 days. So that's about 3 to 4 months. The lifespan of a sickle cell is 3 weeks. See the difference there? 3 months, all right, 3 or 4 months versus 3 weeks. Big difference there. Now, when we say the word anemia, all right, anemia is usually caused by that people do not get enough oxygen to their tissues. And when you're anemic, you're tired, your color might be a little white or ashen, all right. You're fatigued, you're dizzy, all right. It's not enough oxygen reaching the tissues. And it can be caused by a lot of things. It can be caused by not enough red blood cells, but it can also be caused by cells with abnormal structures in it. So here is our little sickle cell. Now the big problem with sickle cell is that because the cells are shaped funny, they can cause occlusions. They can cause blockages in blood vessels. Here's a normal red blood cell, and because they bend and flex, they can actually go through tiny little capillaries quite easily. But sickle cells, because they're stiff, you'll notice that they often get stuck. Especially when there's a lot of them. If there's a lot of them, they can get stuck in all kinds of places. They can get stuck in the blood vessels of a joint. They can get stuck in the blood vessels of the brain and lead to a stroke. They can get stuck in the blood vessels of the heart, leading to a heart attack, or to the kidneys, causing kidney failure, or to the liver, or any other little organ. Right? So these can actually be life-threatening, especially when there's a whole lot of them. Right? Sometimes you'll hear about people who suffer from a sickle cell crisis, and usually what's happened with that is a lot of their red blood cells have turned and sickled. And because they're inflexible, they cause a lot of blockages. And then the person usually has severe pain and all kinds of other little problems to them. So, what makes a sickle cell so stiff? Well, I want to show you a picture, and this picture is really great because it shows the fibers. This is something you only see in a sickle cell. Normal red blood cell, you won't see this at all because it doesn't exist. Right. So let's take a look at it. This is an electron micrograph of a sickle cell. Now look at all those fibers in there. No wonder it's stiff. Right? No wonder it's stiff. It's like putting a bunch of pipe cleaners in a bag, all right? And you have another bag that's just filled, let's say, with ketchup. All right? The one with pipe cleaners is not going to be as flexible as the one with ketchup. Right? So that's what's causing the problem, is this existence of these fibers. So these fibers are weird, absolutely weird. So we need to know what they are made of. So let me show you a cool picture. This is another electron micrograph of just one of these fibers. And here it is. That's what the fiber looks like. Now the cool thing about it is we've taken these pictures and we've colorized them just to make them a little bit easier to see. And so if we take our fiber and sort of break it down and turn it around and look at the front end and keep going, all right? If we keep going, what we eventually start to see is that it is made of nothing but hemoglobin. In a normal red blood cell, the hemoglobin stays separate from each other. They don't bond to each other. They stay separate. But for some reason in sickle cell, the hemoglobin will go and start sticking to each other and make these gigantic fibers that make the cells very, very stiff. Isn't that weird? That the fibers that make them stiff is going to be built from hemoglobin, which normal red blood cells have. So there must be something odd about this hemoglobin that makes it do it this way. Now, how the fibers are made is a cool question. In normal hemoglobin, we often abbreviate hemoglobin as just HB, so that's HB, stands for hemoglobin. And hemoglobin A is normal hemoglobin. When a red blood cell carries its oxygen and it gets down to the tissues and the oxygen leaves the red blood cell, right? The hemoglobin doesn't bond to anything, it stays separate. So these represent hemoglobin molecules inside a red blood cell. So when they give up their oxygen and become deoxygenated, you'll notice that they stay separate. But the sickle cell, and we abbreviate HBS, when it gives up its oxygen, you'll notice that that's what forms the fibers. So as the oxygen is released from the hemoglobin molecules, it makes them sticky, and they form these super long polymers, all right, that form these stiff fibers. So that's the reason why the fibers are formed, is because when they give up their oxygen, the hemoglobin molecules like to stick. Now when they take on oxygen, they're going to split up again. Right? So through that cycle, and it takes about 70 seconds. That's how long it takes a blood cell to circulate through your body, about 70 seconds. Right? So if you think about every pass, all right, it's giving up oxygen, and then it's taking on oxygen. So those fibers are dissolving and then forming back, and then dissolving and forming back, and dissolving and forming back. Now, this isn't much of a problem if you have a lot of oxygen. If you have a lot of oxygen, you're pretty much okay. But if you get to a position where you're in oxygen deficit, let's say you go to a higher altitude. Let's say you decide you're going to go to Colorado and you have sickle cell. With the lower amount of oxygen, that means more of your red blood cells will sickle. Right? If you exert yourself too much, let's say you decide, I'm going to go and run the track, right? And you go into oxygen debt, that means more of your red blood cells will go into sickle. Right? So oftentimes when people have a sickle cell crisis, it's usually from something where they have had an oxygen deficit. They have suffered a little bit of what we call hypoxia, low amounts of oxygen. Because that makes more of the hemoglobin give up their oxygen and makes them form the fibers. Now, turns out it all has to do with a malformation. And the malformation, as you can see, is there in the hemoglobin molecule. So this is normal hemoglobin, and you can see the 4 chains, 4 polypeptide chains. But if you look at sickle cell hemoglobin, you'll notice that it's odd. It's shaped funny. This shape is what causes all the problems. And we can trace what causes this to be shaped wrong down to a single amino acid. That's the amazing thing about sickle cell. It comes down just to one amino acid that's different. If you look at the beta globin chain, that's where the problem is. It has 147 amino acids. Between normal and sickle cell, of those 147, there's only one that is wrong. Everything else is perfectly fine. You just have one amino acid that's different. And we've discovered where it is. It's amino acid number 6. So out of that 147 amino acids in the beta globins, we have one amino acid that's different. You'll notice in the normal one, it's number 6. It's glutamic acid. All right. Now think about glutamic acid. All right. It loves water because it has a charge. So it's going to be fine on the surface of the protein. It's going to be perfectly fine there. Right? Valine, on the other hand, is hydrophobic. Where does hydrophobic amino acids like to be? Down in the interior. So this one is perfectly fine being on the surface. This one wants to be inside. And that makes all the difference. Let me show you this cool picture. So here's normal hemoglobin, and here is the shape. And you can see number 6 is just fine. But look what's happened here in sickle cell. All of a sudden, that particular area, it's trying to get away from the surface. It's trying to go to the interior, trying to get to the center. And this weird shape actually goes and causes it to bind. You can see all of a sudden it's created almost like Lego blocks that interlock perfectly to form our polymer. So that one little amino acid made all the difference in the world on the shape of the protein. That's pretty remarkable. In fact, when we go and take a look at the over 3,000 genetic disorders that afflict humanity, we find that a lot of them are caused by amino acid out of sequence, a protein that's shaped wrong. So if you think about in determining the final shape of the protein, how the protein is going to fold, the most important level of organization in the folding is that sequence of amino acids, that primary structure. So what it tells us that the primary structure, that sequence of amino acids, really dictates how that protein is going to fold. Right? It's the most important level of organization. It reminds me of that saying that, right, the beginning determines the end. Right? So one of the cool things we can do is we can look at proteins that vary from one critter to the next. Right? And we can usually tell, okay, well, that one doesn't work right because it may have something wrong with the sequence. And usually it might be as simple as a single amino acid out of sequence that makes it non-functional or function in a worse way. So all the problem with sickle cell anemia comes down to just one amino acid out of sequence. I wanted to show you one of my favorite websites. Unfortunately, I haven't checked it since the spring, but every time a protein is deciphered, meaning that its sequence has been deciphered, its structure has been deciphered, and someone's published it in a scientific paper, that protein becomes part of a database. We now have a database of all the different proteins that have been described. And it's pretty amazing. In fact, let me show it to you. This is called I'll come back to that one. It's called the Protein Data Bank, and it's been around for a long time. And when I started looking at the Protein Data Bank, probably about 10 or 12 years ago, the number of proteins that had been described was about 35,000. So in about 12 years, they've sort of tripled that, the number of proteins that have been described. As you can see, and this is from spring semester, 100,000 different proteins. Let me just give you a little example of one. Next week we'll be doing a little lab where we examine an enzyme called catalase. And every organism on the planet that uses oxygen has it. So it's it's found among most living organisms. And what catalase does is actually go and break down a byproduct of a toxic form of oxygen. We have a form of oxygen that's called a superoxide free radical that can mess up chemical reactions. And we deal with it by having two enzymes, and the second enzyme is catalase. Catalase actually goes and takes hydrogen peroxide and breaks it down into water and oxygen gas. So every organism that uses oxygen has that one, including us. So I was interested in that particular little protein, and I just typed in catalase, and I found one that actually comes from a little yeast, a little mold there, Penicillium mold. And I thought, oh, that's cool. And as you can see, it tells us when it was described. And you can see the method. Here's X-ray diffraction crystallography. Most of them do it that way. All right. And in this case, they go and show us what the molecule looks like and where the publication comes from. But one of the cool things about it, too, is they have, and this is a little old, but they have all these really cool viewers. Let's say that you're a pharmacist or a biomedical researcher, and you're looking at a drug or you're looking at an enzyme, and you want to be able to look at it from all different sides to see how it will bond. Maybe there's an active site somewhere that you have to take a close look at. Well, you can do that. They have a little viewer that allows you to spin it around and see it from all kinds of different angles. You can look at it in a ball and stick model, a ribbon model, a space-filling model. But one of the really great things about it is it tells you a bunch of information. Turns out this enzyme, catalase, in this little mold is 659 residues. Now you might be wondering, what the heck is a residue? That's just a word meaning amino acids. So this one has 659 amino acids. That's how long this protein is. All right, 659 amino acids. The cool thing is they also break it up into their secondary structures. So as you can see, 23% of the protein is alpha helix and 7% beta sheet or pleated sheet. Now, I love this little picture here because they actually go and show you different little parts. These little parts that you see here that are in yellow, that represents prolines. And if you remember, prolines are where they bend, where the protein bends. So those are our little corners, the little bends of the protein. You can see this little area right here that tells us that between those amino acids, between 41 and 50, there's a beautiful alpha helix. And the blue ones tell us those where pleated sheets are located. So that's pretty cool. It shows us what the secondary structures are, what number the structures begin, and how many different amino acids we got. All right. Cool. Now, I want to show you two little videos, and I'm going to begin with a video that I was particularly happy about because we're actually going to go and make it in lab today. All right. And this one is an examination of a beautiful little protein. And it shows the tertiary structure and also two secondary structures. Now, this little protein has an odd name, but it has a very important function. It's called a zinc finger. That might sound a little weird. It's called a zinc finger because this little protein has a little molecule, a little atom of zinc connected to it. We'll find out that oftentimes proteins will have a non-protein helper or non-protein molecule often attached to them. And oftentimes when they're doing some sort of chemical reaction, they'll often go and have a little metal, and this one does. Now the zinc finger is important because what it does is it interacts with DNA. Right? DNA has genetic expression. Sometimes genes are turned off. Sometimes our genes are turned on. And the zinc finger help with the genetic expression regulation. It actually goes in bonds directly to the DNA. Right? And so they're well studied, and we know a lot about them, but they're a beautiful little small protein that does a very important job. And I want to show you a little video